Plasminogen Activator by G. Manimaran, 2nd MSc Biotechnology. Tissue plasminogen activator is a protein involving the breakdown of blood clots. It is a serine protease found in endoepithelial cells, the cells that cell line the blood vessels. As an enzyme, it catalyzes the conversion of plasminogen into plasmin, the major enzyme responsible for breakdown of clot breakdown. The human TPA has a molecular weight of approximately 70 kilodaltons. The table shows that name, synonyms, history and synthesis, molecular weight, carbohydrate, genes and amino acids and concentration, half-life period, types and functions of the TPA. Blood clots is an enzymatic event initiated by the substance from injured tissues and compelling in the formation of blood clot formation. Fibrin monomers. After a few days, the fibrin clot is degraded by the monolytic enzyme system. A central enzyme component in the system is glycoprotein, plasminogen, present in the plasma and extravascular fluids. The plasminogen is an cymogen of serine protease which, following a partial cleavage by plasminogen activator, is converted in the form of plasmin. Plasmin is involved in a variety of biological processes including cell migration, growth, inflammation and tumor innovations. Although its primary function is assumed to be lysis of fibrinogen into the vascular rays, the two plasminogen activators are found in a human body, the tissue type plasminogen activator and urinary type plasminogen activator. TPA is the principal activator of plasminogenous blood, whereas UPA is the main function of tissue related proteolysis, and it's believed to only to be a TPA and removal of intravascular fibrin. Fibrillolysis is the initiation and propagation is mainly by the fibrin surface, which binding sites are optimal contact between a number of the components of fibrinolytic systems. Most notably, fibrinogen and DPA is a stimulatory effect of ensures a high concentration of plasminogen and DPA at a fibrin deposits and localize that plasmin activity. Incubator regulation is provided by the plasminogen activator BA1 and the plasmin incubators. BAI1 is the most efficient incubator of TPA in plasma, and majority of circulating DPA is bounded with the incubator. Normally, there is existing careful regulatory balance between a formation of fibrin and its subsequent removal. However, in certain pathological conditions, inhibitory deficiency and balancing is distributed and subject with the inherent increasing of fibrinolytic activities. Usually harassing several beadings. In contrast, a decreased fibrinolytic activity may be associated with thrombotic disease. Components of that's a major components of fibrinolytic systems. TPA structures and function. The human TCA is a 68 kilodalton serine protease composed of 550 and 527 amino acids and contain between 7 to 30% carbohydrates. The molecule is made up of 5 distinct domain structures with autonomous functions. The finger domain and 2 Finkel K domains are involved in the binding of TPA to fibrin, whereas epidermal growth factor domain is included in a rapid hepatic clearance of molecules. The serine protease domain containing active site region and made up of serine, histidine and aspartic acid, which is suitably related for part of each other in primary structure but are closely proximate in a folded protein. The region cleavage of arginine 561 and a valine 562 bond in a plasminogen thus activated it to the plasmin. It's the domain structure of DPA. Synthesis and secretion. DPA is synthesis mainly in a vascular and endoepithelial cells and is secreted into the plasma continuously, and also the aquatic release of TPA. Later situations occur upon stimulation of certain endoepithelial receptors, a different region of vascular system secreted and different amount of TPA. Upper estimin secretion about 4 times of more DPA than amount of lower estimins. There are two forms of DPA, single chain DPA and two chain DPA. The single chain molecule is native of DPA secreted from endoepithelial cells, where two chain form is a result of proteolytic enzymes of plasmin. Both forms are catalyst activity and have a similar enzymatic property in the presence of fibrin. Plasminogen activator incubator. A number of incubators have been identified in plasmin and other body fluids with the capacity of incubate TPA and incubating BI1 and BA2 plasminogen activators. BA1 plasminogens the protease nexin and macroglobulin, thesperin incubator and CA incubators. Other pills. Alpplace is a tissue plasminogen activator produced by recombinant DNA technology from human tissue culture. The drug used in lysing thrombus. It is the production and mode of the mode action of TPA. Plasminogen diseases, deep vein thrombosis, cardiovascular disease, and thrombolytic therapies. Reference from diaphragm. Thank you.